DNA is vital for all living things. It's important for inheritance, coding for proteins, and the genetic instruction guide for all of life and its processes. It makes everything what it is. DNA is commonly thought of as the blueprint of life. First of all, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR itself is a family of DNA sequences found within the genomes of prokaryotic organisms such as eubacteria and archaebacteria. CRISPR is involved in a method of gene editing that's based off of a natural system used by bacteria to protect themselves against infection from viruses. The first hint of CRISPR's existence was in 1987 at Osaka University in Japan. Researcher Yoshizumi Ishino and his colleagues accidentally cloned part of a CRISPR sequence during an analysis of the genes involved in phosphate metabolism in E. coli bacteria. They noticed that the organization of DNA repeats was unusual. Repeated sequences are usually arranged consecutively without interspersed different sequences. Little did they know that they were looking at a huge advance in science, something that has the capability to change the world and life as we know it. How does this gene editing technology actually work? Well, it involves a super important nuclease enzyme called Cas9 that functions like a pair of molecular scissors for DNA. Together, CRISPR and Cas9 have the amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning, and then they can disable it by cleaving the DNA. To use CRISPR-Cas9, we must first identify the gene that we'd like to work with. For Cas9 to find that specific gene, we must attach it to a guide RNA sequence that matches the specific gene we want to edit. Cas9 and the guide RNA bind to the desired sequence and cleave it with a chemical reaction. From there, we have the ability to modify the existing gene, insert a new sequence, or delete the sequence altogether. It's like DNA cut and paste. What does this mean to the rest of the world? Well, CRISPR-Cas9 has a wide variety of potential applications. It can be used in basic biology research, the development of biotechnology products, and the treatment of diseases. CRISPR-Cas9 technology is easy to use and is about 4 times more efficient than the previous best genome editing tool. So far, CRISPR's biggest effect has been felt in basic biology labs, but CRISPR technology has been applied in the food and agricultural industries to engineer probiotic cultures and to vaccinate industrial cultures. CRISPR-Cas9 is also being used in crops to improve yield, drought tolerance, and nutritional properties. Another potential application is to create gene drives. These are genetic systems which increase the chances of a particular trait passing on from parent to offspring. Eventually, over the course of generations, the trait spreads through entire populations. Gene drives can aid in controlling the spread of diseases such as malaria by enhancing sterility among the disease vector, in this case, female mosquitoes. In addition, gene drives can also be used to eradicate invasive species and reverse pesticide and herbicide resistance. However, gene editing does not come without its drawbacks. There's also the phenomenon of off-target effects, where DNA is cut at sites other than the intended target. This can lead to the introduction of unintended mutations. CRISPR-Cas9 brings up some ethical issues. Should we make changes that could fundamentally affect future generations without having their consent? What if the use of gene editing veers from being a therapeutic tool to an enhancement tool for various human characteristics? Now here's a little bit of math. Science equals knowledge, knowledge equals power, and power equals responsibility. If we're going to change the world, are we going to do it for the good or for the bad? It's our choice.